Hi there, and welcome back to another video in our Summertime Science video series. My name is Chris Harvey, and I'm a senior lecturer in biology at Canterbury Christ Church University. And today's video, I'm going to take you back into my garden, and we're going to look for a really cool microscopic worm that lives in the soil. Now these worms are insect-killing nematodes. So that means they find an insect, they invade the insect, they crawl inside their body, they vomit out deadly bacteria, the insect dies, the worms eat the insect from the inside out, and then thousands of new worms emerge to go out into the world to find a new insect to kill. Sounds really creepy, right? Luckily they're microscopically small, so they won't attack humans or anything like that. But let's see if we can find some of these worms in my own garden, and you can go on a hunt to find them in your garden as well. So let me show you what you'll need to do that. Here, laid out on the table in front of me, I have the various different bits of equipment I'll need for today's experiment. Now, these two things over here I'll mention in a moment. They're actually not essential for the experiment, so you can do without these. But what you will need is some kind of container. What really works well are like these clear sandwich bags or Ziploc bags, anything like that, food bags. You can use old ones, don't have to use fresh ones. If you want to save on plastic, just ask your parents or find some in your kitchen or something. Find some nice sandwich bags to use. You'll also need a trowel. We're going to be digging up some soil because, like I mentioned, these worms live in the soil. So you'll need something like this. You will also need some kind of insect to bait your sample with. So we're going to take a soil sample and then we're going to what's called bait it with an insect. And the worms are hopefully, if they're in the soil, are going to find the insect, they're going to infect it, and that's going to show us that they're there. So the insects I have here, let's turn this over, they're called waxworms. You can see them here crawling around. They're not harmful, they don't bite, they're not, they're little squidgy worms, and these will turn into moths when they're adults. You can buy these as pet food on the internet. So this box here cost me about £10, but you can get smaller ones that are sufficient for your experiment for about £4 or £5. You can also get them at Pets at Home, for example, in pet shops, because they're used as lizard food. Okay, so let me tell you about these two bits here. These are optional. You don't need those for the experiment to work, but they're useful as what's called a positive control. So a positive control is where we try to set up an experiment in a way that we know we're going to get positive results if everything works out right, so that helps us to confirm that the experiment was done correctly. So what we have here is just a simple watering can. I've got about 8 or 10 liters of water in there. And what I have here is a box of these insect-killing worms I'm going to be looking for. This is a brand name. They're called Nemesis. You can see they're a biological vine weevil killer. So these are sold as biological pest control. So if you have insect pests in your garden, you can buy some of these online. This one cost me about £12. You can see inside here, let's get this out. It has a little box, a little container that is full of these worms, and they've been dried. That's how amazing these worms are. They can survive being dried out, packaged like this, and then if I put them back into the water here, they will come back to life. Amazing stuff. Now you can see here it has a use before date. Today's the 17th, or 18th even, no, 17th. So we're a bit over the use by date, but hopefully we'll still get a positive result that's going to confirm for us that the experiment worked. So what I'm going to do is I'm going to pop these guys into the watering can. I'm just going to open up the pack here and then chuck them all in there. There we go. That's most of them. Take the rest out. Okay, and it says on the pack here that it's about 6 million worms. So 6 million worms, that is a lot, obviously. And so that should definitely be something we should be able to detect with our experiment. There we go. So these worms we're looking for are called Entomopathogenic Nematodes. So in Greek, that means insect-killing nematodes. They're insect pathogens, essentially. They're tiny. They're about 1 millimeter in length when they're fully grown. And the free-living stage, the one you'll usually find in the soil, is the juvenile stage. And the juvenile stage is the one that goes out foraging to find an insect host. It invades the insect host, burrows inside the insect host body. It has a symbiotic bacteria it carries with it that it vomits out into the insect's body. And those bacteria then start to grow and multiply. They're like a bacterial infection inside the insect. And they're strong enough to kill the insect. And once that's happened, the nematodes will start feeding on the bacteria. So they've almost like grown their own food inside the insect. And they will grow and mature become reproductive, lay thousands of eggs, and then you'll have thousands, even hundreds of thousands of new nematodes emerging from the dead insect cadaver to go out and find a new host. So let's see if we can find some of these worms in the soil in my garden. So as I mentioned, the worms we're looking for live in the soil. So they're inside the soil, so what we're going to have to do is dig up a soil sample to see if we can bait it with our insects to find the worms. So what I'm going to do is just go around my garden. Here, for example, I have a nice little flower bed with some nice rich soil. So what I'm going to do is I'm going to just take my trowel, then I'm going to just dig into the soil to about 10 centimeters deep. A bit deeper than that. There we go. And that gives me a nice little soil sample that I can now pop into one of my sandwich bags. So I've got my soil sample right here. As you can see, it doesn't fill the bag completely. So what I'm going to be doing is I'm going to be going around the front garden, sampling various different spots, several spots here in the front bed, but also other nooks and crannies here and there, because different spots, different types of plants and so forth might create soil conditions that are favorable or unfavorable for the nematodes. So I have to take a nice random sample from around the whole front garden, and I'll do the same thing for the back garden and build up a nice little set of samples for us to bait. So once I've done that, I'll show you how to do the baiting process. Now that I've taken a soil sample from this part of the garden, I'm going to try out a positive control. So that means I'm going to take my watering can with my nematodes inside that I've bought. So I know there should be millions of nematodes in here. I'll use that watering can and I'll water the same spot of soil that I've previously taken a sample from. So let's see, here we go. I'm going to put some nice dollop of water in here, and I'll water the rest of the front bed here to spread out these worms very nicely. Now I should say, I'm not doing this in the ideal conditions. These worms are quite sensitive to heat and to UV radiation, so I really shouldn't be pouring them out in the heat of afternoon sun. But that being said, we're just doing this for the experiment, and if I take my sample promptly, I shouldn't have any problems with retrieving some nematodes from this. But if you want to buy these nematodes or these worms to protect your own crops in your garden, for example, or protect lovely flowers in your front bed, then you should apply them ideally just after some nice rainfall on an overcast day. Here's my positive control in a nice separate bag. Now, as you'll see, if I go in close, you can't really see the worms with your naked eye. They're too small, about a millimeter in size to actually see, but they're hopefully going to be in there, and we'll be able to find them once we bait the samples with our insects. Now, the best practice if you're doing an experiment like this is to make sure that you clean your trowel after every sample you take, because if you don't do that, you might be transferring worms from one spot in the soil to another, from one sample to another, and that might give you a false positive in one of your samples. So if you're taking multiple samples, wash your trowel in between samples. So here I am, and as you can see, I've got 1, 2, 3 nice samples to bait with insects to see if I have any nematodes, any worms in them. This here is my positive sample, the one that I know worms should be inside because I've applied some from my watering can. And these two are the samples that I'm going to have to check if I have any naturally occurring worms in my back garden. So how do we bait these with the waxworms? What you want to do is just take a few of them, and again you can handle them with your fingers, not a problem. They're safe to handle, they don't bite or anything like that. And you want to collect, because we have rather big samples, you can put 10 or 20 of those waxworms into one of these bags. The more insects you have crawling through the soil, the more likely one of them will be found by one of the worms and infected, and then you'll be able to see confirmation that you have the worms in your sample. So what I'm going to do now is I'm going to pluck out some of these waxworms, and I'm going to chuck them into these bags, and yeah, I'll catch up with you once I've done that. As you can see, I've got all of my samples nicely bagged up and labeled, and I've added my waxworms to each of the bags. These will now hopefully crawl into the soil and be found by any worms that are in the soil samples. Now, you want to store these for about 7 days in a dry and not too warm, not too cold place, so around 15 to 20 degrees. So if you want to keep them outside, that's fine, but keep them out of the direct sunlight. if you want to store them in the house, just put them in, you know, into a bathroom or a you know, the basement or something like that, where it's nice and, you know, not too hot, not too cold, and there's no direct sunlight falling on them. After about a week, if there are any worms in the samples, they would have infected the insects. So you should check the samples in a week. You can do that by simply pouring out each sample into a tray and then looking for signs of infection in the insects that are in the sample. Now I'll put up some pictures here. If your insects turn black, then that's not a worm infection. Because the worms, the bacteria they actually release into the insect, those bacteria produce antimicrobial agents. So they're actually trying to keep out other bacteria from infecting the insect. It's amazing, isn't it, right? So the worms have a way of protecting their resource, protecting the insect from secondary infection. So the species that we're looking for, well, the worms are found all over the world, so you can find them in North America, South America, Europe, Asia, all over the world. But the species we have most commonly in the UK is called Steinernema feltiae. So that species causes the insect to turn a creamy, grayish, brownish color, somewhere in that range. So if you find an insect that's nice and limp and has a gray color or a creamy caramel color, that could be a worm infection. So what we'll do is, in a week from now, I'll do another video where I catch up with you and show you how I look for the infection in the insects, and then you can see if you found any worms in your own samples. So see you next week, and have fun taking samples.